Our focus on this video shall be on the nucleic acid. We'll discuss the general structure of nucleic acid, the helical strand of RNA and DNA. We discuss the complementary base pairing in DNA, and also we'll talk about purine and pyrimidine. So now let's go. When you talk of nucleic acid, you are just describing using a general term for both deoxyribonucleic acid and ribonucleic acid. So that's the general term for DNA and RNA. Yes, so DNA and RNA. So they are one of the biomolecules we have in our body. They help to create and they encode and store information in our body. For example, now the information for the color of our skin they are created by DNA. Are you getting it? They are created by DNA. The information for the color of our skin. Now the nucleic acid, is present on the chromosome. Are you getting it? So the chromosome itself is located inside the nucleus of a cell. So now let me quickly follow my guide here. Let me talk about the general structure of nucleic acid. So now nucleic acid is made up of monomers, and the monomers of nucleic acid are nucleotide. Right? So the same way we have in protein, we have amino acids. In carbohydrate, we have simple sugars. So the same way for nucleic acids, the repeating units are the nucleotides. Okay, let me write that down. Nucleotides. So these are the repeating units of nucleic acid. Now let's go. Now when you talk of nucleotide, what are you talking about? Now this nucleotide, in it we have a 5-carbon sugar. In it also we have nitrogen base. In it also, in the nucleotide also, after the 5 we have a phosphate group. I get it. So that's what the nucleotide is made up. So let me quickly show you the structure of nucleotide, how it really looks like. Now this is the let me give you a sketch of it. Now I said we have a 5-carbon sugar. Now this is the 5-carbon sugar. Oh, okay. Let me put this. This is 5-carbon sugar. So now this is the 5-carbon sugar. So okay, let me put this. Now this sugar is what we called ribose. Are you getting this sugar I'm drawing here? Is called ribose. Now this is a ribose. This is ribose sugar. Are you getting it? So now sometimes you can have a deoxyribose sugar. Now how does a deoxyribose sugar look like? So there will be no oxygen here on the second. Now let me quickly show you something. This is the first carbon here at this joint, first carbon. Another carbon here, carbon 2 here. Another carbon here, carbon 3 here. Another carbon here, carbon 4 here. Then here we have carbon 5. So let me label it 1, 2, 3. This carbon 3. This carbon 4. This carbon 5. So it goes on and on like that. So this ribose. Okay, sorry. So now when you remove the oxygen, as I said, then what you have is deoxyribose. Now it goes on and on like that. Now so let's continue. Now I mentioned something that is made up of a 5-carbon sugar. Now this is the 5-carbon sugar. Now followed by the base. Are you getting it? Followed by the base. Now this is the base. Where you have hydroxy group here, the hydroxy group will be removed. Then you have the base here. Now nitrogen base. When you're talking of nitrogen base, we have 5 nitrogen bases. We have thymine. We have adenine. So these are nitrogen bases. We have cytosine. Then finally we have guanine, and we also have uracil. So these are the nitrogen bases we have. Now at the carbon 1 here, nitrogen bases are tagged. So don't forget we are describing the structure of nucleotide. So now we have, let's say we have adenine here. Okay, let me just put nitrogen base. Let me represent with base. The base is just a general term. It could be adenine, cytosine, it could be guanine or uracil. So now let's go. Now finally, then it also has a 5-carbon sugar, this is the sugar, a nitrogen base, this is the base here, then a phosphate group, Now, do you know the same thing that happened here for a phosphate group? Now this is a phosphate group. Let me show you the structure of it. This is a phosphate group. Okay. Can you see this is a phosphate group? Now a phosphate group will attack here. It will also chase away the OH here. So when it chases it away, so what you have finally is this. You have O. So that's it. P. So also negative there, then double O. So now this structure you have here is a nucleotide. So this is what we call nucleotide. So these three mixed together is what we call nucleotide. Now let me quickly show you. There's something called nucleoside. So nucleoside is just the sugar plus the base. That's nucleoside. I should have shown that before drawing the. So now this nucleotide, now this is the base part of it, this is the sugar part of it, and this is the phosphate part of it. Now let me show you what a nucleoside is. Now this nucleoside, when you have HO, so just the sugar and the base make up the nucleoside. So now that we have done that, we can proceed to the other parts of the study. Now let's move to the helical strand of RNA and DNA. Helical strand of RNA and DNA. So now, let's take care of that. So now let's go. Helical strand of RNA and DNA. Let me quickly mention to you that DNA contains 2 helical strands, while RNA contains just 1 helical strand. So let's go. Let me first do for RNA and DNA. So now we have this for I think it's okay like this. So the same way, let's have it here also. So now this RNA, why this is DNA. So let's go. So now this one. Let me stop there. So the same thing goes here also. So let's do it like that. So this is our DNA. This is the helical structure of each of them. Now what are we talking when I talk about the strand? This one strand on its own, this is another strand here. So this one has only 1 strand, can you see? So that's the difference between the DNA and RNA. So we can now move forward and continue and discuss the complementary base pairing between the two.